Yeah, 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 yeah.
Okay. IRS signalosome functions are dependent on LLPS formation. Okay. So insulin and IGF-1 are signaling molecules which regulate many metabolic cellular activities, such as glucose uptake, as shown in this diagram below. The most well-known effect is that the GLUT4 glucose transporter will be upregulated on the cell surface, allowing for glucose in the blood to be taken into cells. This process occurs within minutes. The lack of response to the insulin signal is implicated in things such as type 2 diabetes. There are many ways in which this lack of a response can occur along the signal transduction pathway. Take a look at the signal transduction pathway on the diagram. You got a couple of nodes here. You got the first node composed of IRS proteins, which are insulin receptor substrate. It's a family of proteins. And then that signal gets transduced to node two, which is PI3 kinase. And then lastly, third node, AKT. And then bada bing, bada boom, you get stuff in the cell happening, such as glucose uptake, glucose synthesis. You can see it all below. So insulin and IGF signals are very similar. You can see that there's quite a bit of overlap. In this study, we're just going to focus on IGF signals. So yeah, this first node here, we're going to be looking at IRS-1 specifically, but they're all very similar. Boom. So that first node of IRS proteins is thought to be a signalosome. What is a signalosome? Signalosomes are phase-separated protein hubs which promote the speed of cell signaling. They are characterized by high protein concentration. They are membraneless organelles and have liquid-like properties which allow for sensitivity to the strength of the signal they receive. These properties also allow them to interact efficiently with their effectors, which are often dispersed amongst the cell or have low affinity. So LLPS increases that affinity. It's more sensitive. It's often required for signaling, and this was proven in a study where they looked at T cell receptor signaling and WNT signaling. So IRS-1 has three domains. It has a pH domain and a PTB domain. That's what's shown in blue in the diagram right there. Since previous studies have linked IDRs to inter and intra-protein interactions, which are essential for LLPS formation, Gao et al. In a paper titled Phase Separation of IRS-1 drives the formation of insulin slash IGF-1 signalosomes. They hypothesized that the CTD of IRS-1 may function similarly. In doing so, they not only prove that the IDR of IRS-1 is responsible for LLPS formation, but also what affects this. This is the paper. This is the paper right here. That one right there. That one on screen. The general approaches used in this study. First, they set up a doxycycline inducible cell line for this fusion protein of IRS-1. Attach GFP to it, induce it with doxycycline. Pretty simple. They also checked that upon induction, it expresses at similar levels to that of a normal cell. Um, the general workflow of this experiment begins with identifying the distribution or presence of fluorescently tagged proteins using confocal imaging. Co-localization and or LLPS formation can be assessed this way, and the authors usually follow these observations up with immunoblotting to determine if binding is occurring between LLPS and or co-localized components. Additionally, FRAP and DIC are used to assess the dynamics of LLPS molecules. They're sort of besides the point. They're just quantifying the LLPS in that way. So in figure one, the authors have built off of their previous research topics in this top left panel here, where they looked at RAB5. RAB5 was recently discovered to promote myoblast differentiation and directly interact with IRS-1. And here they've just shown that again. They showed co-localization. See where my cursor is. You can see that they form circular globs, raising the hypothesis that IRS-1 undergoes phase transition. They then identified which regions of the protein are responsible for the globular arrangements by making new GFP fusion proteins of IRS-1 portions, one for the PH-PTB domains and that for the IDR. As you can see right here, IDR, but not the PH/PTB portions result in globs. Uh, they quantify these globs using FRAP. You can see that they fuse and that they also recover quite quickly, suggesting that these molecules are quite dynamic. Here, the authors ask exactly what is responsible for self-assembly. They must be part of the IDR because of the last figure, where the IDR was shown to form circular gloves, but not the PH and the PTB truncated portions of that protein. So now they made various truncation mutants of the IDR region and they flag tagged it. They then co-infected these mutants with the IDR GFP fusion IRS-1 proteins. And when performing the immunoblot by probing for GFP, it was qualitatively assessed whether or not the flag tagged truncation mutants were binding or not. So you can see that the 301 to 600 AA region is pulling the full length IDR out, and a little bit the 801 to 1000 region as well, but to a lesser extent. They called this 301 to 600 region SAR, self-assembly region. They then analyzed each IRS-1 truncation mutant in vivo using confocal imagery, showing that the deletion of the SAR region reduces the amount of globs compared to the wild type. All right, knowing that IRS-1 is phosphorylated upon insulin signaling, the authors wondered how phosphorylation affects LLPS formation. It has been previously shown that the IDR is home to many tyrosine phosphorylation sites, which can serve as interacting motifs for SH2 containing effectors such as P85 or GRBT. GRB2, which are regulatory subunits of PI3 kinase. So it was hypothesized that the IRS-1 droplets could act as signalosomes, enhancing, regulating, and facilitating signal transduction. So they wondered if IGF stimulation would affect LLPS. This figure right here shows what happens in IGF starvation and IGF stimulation conditions. You can see that LLPS only forms when stimulated. Additionally, tyrosine 608 gets phosphorylated in IGF-1 stimulatory conditions. This is important because the principal site of interaction with P85 and the interaction of PI3 kinase is through this residue. So to assess how critical these phosphorylation events are to IRS signalosome assembly, they remove the ability for nine tyrosine residues to be phosphorylated by mutating them to alanines. And the consequence of this is shown by confocal imagery again in figure 4I, right here. You can see the number of LLPS has reduced drastically. Figure 5. How is IRS LLPS formation relevant to insulin/IGF signaling pathways? If IRS is known to interact with P85 for downstream signaling, IRS-1 LLPS formation may be critical for mediating downstream effectors. So they tested this. This figure shows confocal images of endogenous P85 and IRS mutants in starvation and IGF stimulated conditions. This is the figure. Bada bing, bada boom. So removing the ability for LLPS to form by blocking phosphorylation of the IDR or deleting the SAR or starving the cells of IGF results in no colocalization. Therefore, no signal transduction, in theory. Figure 6. Previous studies of IRS-1 SNPs have shown associations to metabolic diseases. The authors wanted to see how specifically the G972R SNP, one of the most common IRS-1 SNPs, affects LLPS formation. They took the exact same experimental setup shown in the previous figure, but compared wild type IRS to the G972R mutated IRS. And found reduced LLPS formation and consequently reduced P85 colocalization. Boom, look at that. See, look at these. So only when IGF is provided and the full-length protein is available do these two proteins colocalize. And then they confirmed this with immunoblotting. Um, they followed these results up with immunoblotting with P85 and GRB2, another P1 PI3 kinase subunit, and found data supporting the idea that this SNP is reducing the efficiency in which IRS can recruit downstream effectors. Notice that this mutation doesn't completely kill IRS-1's ability to recruit downstream effectors because figure 3 established that the 301 to 600 amino acid region is most importantly for self-assembly, and the 800 to 1,000 amino acid region has a smaller contribution, and that's where this SNP is located. So it doesn't completely kill the ability slash functionality for IRS to form LLPS molecules. Discussion. So here's the main takeaway from this paper. The self-association of IRS-1 into dynamic LLPS molecules is critical in IGF signal transduction. The lack of a membrane, high local protein concentration, and liquid-like properties are all aspects of signalosomes which allow for robust signal transduction and recruitment of PI3 kinase to take place. The importance of tyrosine phosphorylation in the IDR of IRS-1 in self-assembly of the IRS-1 signalosome was uncovered. And may explain the pathological effects observed in type 2 diabetes alongside that G972R mutation. So some questions arise from this paper. Uh, so some questions arise from this paper. The IDR of this protein was found to clearly have a function, right? So this may help shed light on the functionality of IDRs and other proteins. So what other proteins have IDRs that may also function in this way? Do we know if they have similar function? Do we know this? I don't know. It's a question for future research. Um, what other pathways are regulated by the formation of LLPS? It's already found that T cell receptor and WNT signaling pathways work like this, and now IRS same way. Well, there's got to be more. You got to think there's got to be more. Uh, there also may be other post-translational modifications other than phosphorylation that affect LLPS formation. What other layers of control could possibly exist? Um, and can we alter these? Now that we know that G972R mutants have a reduced capacity to form LLPS, is there any way we can restore that? We know the problem. Can we identify a solution? Can we find a solution? Is there a way to reverse this? Farewell.